Good morning, class. Today in this lecture talk, I am discussing with you BSc third year paper 5 and paper 6 BCU practical scheme. Before I begin today's presentation, today's thought, be sincere, not serious. Okay. Now let us go through the question pattern of paper 5, environmental and immunotechnology of BSc 5th semester biotechnology course. Here for this experiment, duration 3 hours, maximum marks 30. Question number 1, estimate the total hardness of the given water sample. Okay, this question carries 8 marks. Okay, this 8 marks we are splitting in the scheme of evaluation as performance 4 marks. How we are performing everything that carries 4 marks. Then principle 2 mark, calculation and result 2 marks. So overall together you have 8 marks here. Then question number 2, prepare a temporary slide of VAM or rhizobium. So VAM sample will be given. We need to focus and observe under the microscope. Okay, prepare the slide of the VAM, focus, observe the microscope, and then call the examiner. If your slide is good, they'll give marks for your performance. Or else you can also you may get with the rhizobium. Okay, rhizobium temporary slide you need to prepare by doing gram staining. Okay, here this carries 5 marks. Here the 5 marks will split into performance 4 marks, then result 1 mark. Okay, total 5 mark. Then question number 3, perform the differential count of WBC. Okay, this question carries 7 marks. Here in this question number 3, performance 3 marks, calculation result 4 marks. Again, for this question number 2, there won't be any principal procedure. Okay, you need to just give the calculation and result. Then question number 4, perform RPR or Vidal or ABO blood grouping from the given sample, and this question number 4 carries 5 marks. So in the examination hall, the external examiner may give you RPR, rapid plasma reagent, or VDRL, then Vidal, okay, which is for the typhoid test, RPR for the syphilis test, then ABO blood grouping to check whether the person belongs to which type of blood group. Okay, in these 3, any one experiment you are going to perform, and this carries 5 marks. Here in the question number 4, 5 marks, a scheme of evaluation. Here principle 2 marks. Okay, what you write the principle regarding RPR, Vidal, ABO blood grouping carries 2 marks. Then performance and result carries 3 marks. Okay, this is about question number 4, which carry 5 marks. Then the last one, Viva O's. Viva O's carries 5 marks. Here, Viva O's, here questions related to theory and practical syllabus of this paper only. Means from environment and immunotechnology theory paper, you may be asked by some questions by the examiner. Okay, then at the end, question number 6, it carries 5 marks for your class record. So submission of the class record is very, very important. Hope you understood about the paper 5, environment and immunotechnology practical scheme of evaluation. Okay, don't be lazy. Okay, so be active and focus on your practical scheme. Then 5th semester biotechnology paper 6, plant and animal biotechnology, maximum marks 35 and duration that is 3 hours. Okay, here question number 1, isolate the protoplast from the given sample. Okay, so this is mandatory, no excuse here. This carries 7 marks. 7 marks we are splitting the scheme of evaluation as performance 3 marks, principle and procedure writing 3 marks, result carry 1 mark. Okay, second question, there is no option again. Isolate and stain the parenchyma cells from the given sample. Okay, so this carries 8 marks. Here again, performance 4 marks, principle and procedure writing part 3 mark, result 1 mark. Okay, Question number 3, prepare synthetic seeds from the sample provided, which carries 4 marks. Performance 2 marks, comment 2 marks. Okay, so this carries 4 marks. Then question number 4, spotters. Okay, you may be asked spotters from the plant biotechnology theory concept or animal biotechnology theory concept. So 2 spotters will be kept over there. Okay, spotters. 2 spotters will be kept over there, and each spotter carries 3 marks. Okay, our 3 into 2 is here. So if we keep 3 spotters, means each spotter carries 2 marks. So total 6 marks. Then what are the spotters you may get in the examination? Here, in the spotter, identification carries 1 mark, relevant point carries 2 marks. So one spotter carries 3 marks. It means we are going to keep only 2 spotters, one from the PBT, another from the ABT. So each spotter carries 3 marks. So total 6 marks, 3 plus 3, 6 mark. So what are the spotters you are going to get here? Spotters here, photographs of callus culture, anther culture, embryo culture, somoclonal variations, somatic hybridizations. Okay. Then other spotters from the ABT, these are the spotters from the plant biotechnology. And from ABT spotters like EGF, FGF, PDGF, serum, BSS, rocks bottle, roller bottle. Okay, so these are the some of the few listed spotters. From these listed spotter, any one will be kept here. From PBT, any one spotter, and from ABT, any one particular spotter will be kept over there. You need to first identify in front of the examiner, which carries 1 mark, and comment on that particular whatever the identification you have given. 11 points you need to write, that carries 2 marks. So at the end, there'll be a viva os, which carries 5 marks. Okay, here questions related to theory and practical syllabus of this chapter will be asked to the students. Then question number 6, that is class record and report. Okay, report, that is nothing but the field visit report, class record and the report. This carries 5 marks. Total 35 marks for your practical experiment. Okay, I hope you understood about this paper 6, a plant and animal biotechnology. Don't be lazy. Please focus on your studies and okay, score good marks without fail. Start studying. Don't sleep. Thank you. Hope you are happy. Best of luck. Stay blessed. You be the gift to the world. Thank you everyone for watching this video.